timeline of the year 2021 to 2022. First, we did the Venom Lab. In this lab, we learned how venom kills, gets into the bloodstream, and how it can affect your blood cells. Our question was, how long does anti-venom take to activate? How venom kills, destroys living cells, causes intense pain, fast-acting blood clots, stop your heart, and bite sting. Procedure steps. Able safety equipment. Go through steps before beginning. Be careful of your surrounding and making sure not to spill anything. Variables. Independent was temperature. Dependent, how long it takes for antivenom to work. Constants and controls. 1 milliliter venom, 1 milliliter antivenom. 2 full blue chemical, and same test tube. Next, we did crime scene lab. We learned about the different search methods of each scene. Link method requires the investigation to be able to form local links between the crime scenes, activity, and the likely places additional evidence could be found at the scene as a result. Line or strip method uses on large outdoor crime scenes. Members of the search team are arranged at regular intervals using arm's length and then proceed to search along straight lines. Grid method used on large outdoor crime scenes. Follow the first line pattern and search in the same manner as the line method. Once the first line pattern is complete, searchers region on the other line pattern. Zone method used on crime scenes that are conspired of readily definable zones such as a house or buildings. Teams are assigned small zones for searching, and then other appropriate searching methods are employed in each zone. Spiral method used on a crime scene with no physical barriers such as open water, can either begin at critical point of the crime scene, outdoor spiral, of the outer edge of the crime scene, inward spiral. Wheel or ray method used on small critical crime scenes. Investigators start from a critical point and travel outward along many straight lines through this point. Next, we did blood types and alleles. We learned the effects of dominant and recessive genes and how that has an effect on the type of blood of offspring would end up having. We also learned with blood types the different types of antigens that go with different types of blood types. For instance, A goes with A or AO, B goes with BB or BO, AB just goes with B or AB, and O goes with OO. The alleles are A is dominant, B is dominant, and O is recessive. Next, we did digestive system. In this lesson of the year, we learned how different animals' digestive systems digest foods differently and the size difference between them. Question: how does the length of the digestive system in rabbits and cats support their absorption of nutrients? In this, we also learned the pH levels of the digestive system, with stomach 1.5 to 3.5, small intestines 6 to 7.4, large 5.5 to 7.5, and the oral cavity 6.5 to 7.5. We also learned the lengths of the animals to see which one of the biggest to smallest using rabbits, a rabbit, a rat, and a cat. heart board games. By using the information or research discussing in class and writing down notes about the heart, we use that information to make a board game that people could be played, have fun with, and have them understand the heart a bit better. Heart dissection. Here, with your dissecting, we learned the different names of the areas of the heart, different tissues, and how it works all together. We learned that the heart is about the size of your fist. Your heart's electrical system controls the rate of the rhythm of your heartbeat. Heart supplies your body with the right amount of blood at the rate needed to work well. The chambers are separated by the heart valves, and any problem with the electrical system can make it hard for the heart to pump blood. The three different tissues: endocardium, the thinner layer of the heart; myocardium, the thick middle layer of the heart; and pericardium, the sac that surrounds the heart. Next, we did transcription versus translation. Transcription is the synthesis of RNA from a DNA template where the code in DNA is converted into the complementary RNA code. Translation is the synthesis of protein from mRNA template where the code in mRNA is converted into an amino acid sequence in a protein. With this lesson, we learned how DNA is replicated and how it's broken apart and made into a new DNA strand. Next, we did diabetes. With learning about diabetes, we learned what causes diabetes and what the differences are between normal people with diabetes type 1 and type 2 diabetes. In normal patients, the pancreas releases insulin, which is then connected to the insulin receptors. Glucose, or C6H12O6, goes through what's called a channel protein. Type 1 diabetic, before the insulin is released, a white blood cell comes and attacks the beta cells, resulting in no insulin being released to the bloodstream. Type 2, no insulin is released, and the insulin receptors are misformed or stop working. Type 1 and Type 2 result in taking insulin as a liquid injected into your body. Next, we did weightlifting. With this experiment, we learned how muscles extension and reflection. We also tested how much weight a person could lift with 0 to 100% effort. We took our resting heart rate before lifting, then took our heart rate after each set of 10 lifts and calculated the difference. With that, we also asked each of the lifters how much effort the last rep took on a scale of 0 to 100. We also learned how oxygen gets to our muscles with each rep and that it's important to breathe with each one. BMI calculations. In BMI calculations, we learned how to calculate BMI and how to categorize the different measurements for BMI. By using the formula, we collected our weight and put it into the formula and saw where we landed on the spectrum.